Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457732. 2. Biological molecules. The biological molecules are based on carbon. Carbohydrates, lipids, and proteins are assembled from organic molecules, e.g. glucose, amino acids, glycerol, and fatty acids. Life is not possible without water, and so understanding of extraordinary properties of water molecule are very important. 2.1 Food tests identify different organic molecules. Organic molecule reagent: initial color, final color. Reducing sugars: Benedict solution, blue brick, red PPT. Non-reducing sugars: HCI and Benedict solution. Blue brick, red PPT. Starch iodine solution, yellow brown blue black. Protein biuret solution, pale blue purple. Lipids: ethanol and water, emulsion test. Clear milk, white suspension. 2.1-1 tests for reducing sugar, starch, lipids, and proteins: Benedict's test, iodine test, ethanol emulsion test, and biuret test. 2.1-2 Semi-quantitative Benedict's test: if reducing sugars are present in foods, Benedict's solution will turn from blue to a brick red precipitate. Benedict's reagent can be used to test for reducing and non-reducing sugars. The test is semi-quantitative on the basis of the color as shown in the tubes. 2.1-3 Test for non-reducing sugar. Benedict's test is used to determine if the sugar sample is non-reducing or reducing in nature. If Benedict's solution is added to the sugar sample and we see no change in the color, then the sugar sample is non-reducing. And if we see a change in the color of the sugar sample, then it is reducing. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457733. 2.2 Carbohydrates and lipids. Carbohydrates contain the elements carbon, hydrogen, and oxygen with the general formula CH(H2O)Y. They are the first products made by the plants in photosynthesis. Most polysaccharides are to glucose. Starch is used for storage in plants, glycogen is used for storage in animals, and cellulose gives strength to plant cell walls. Their three main functions are: main respiratory substrate, e.g. glucose. Food storage compounds, e.g. starch. Structural, e.g. cellulose. Some information about carbohydrates. Type of carbohydrate: examples made up from biological importance: monosaccharide, triose intermediate product in the biochemical pathways of respiration and photosynthesis. Pentose, ribose, deoxyribose. These sugars are found in the nucleic acids RNA and DNA. Hexose, glucose, fructose. Important source of energy in respiration, found in many sweet-tasting fruits. Disaccharide, maltose, sucrose. Maltose. Lactose. Glucose plus fructose. Glucose plus glucose. Glucose plus galactose. The form in which the carbohydrates are transported in plants. Formed from the digestion of starch. The carbohydrate found in milk. Polysaccharide: starch. Glycogen. Cellulose. Glucose. 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 The main storage carbohydrate in plants. The main storage carbohydrate in humans and other animals. An important component of plant cell walls. The basic units of carbohydrates are monosaccharides, characterized by the number of carbon atoms, e.g. pentose sugars have 5 carbons. Starch is a polysaccharide composed of amylose and amylopectin. Its main function is as a storage carbohydrate. Starch and glycogen are suitable for this function because they are insoluble and compact. Cellulose if formed by the linking of beta-glucose molecules. Carbohydrates all contain carbon, hydrogen and oxygen with the general formula CX, H2O, Y. Their three main functions are, main respiratory substrate e.g. glucose food storage compounds, e.g. starch structural, e.g. cellulose. Triose sugars. 3 carbon atoms, e.g. phosphoglyceraldehyde formed in the light-independent stage of photosynthesis. Lecture notes for AS Level Biology by Mohammad Rashid. Chat the cell number 03004610455734. Pentose sugars. 5 carbon atoms, e.g. ribose in RNA and deoxyribose found in DNA. Hexose sugars. 6 carbon atoms, e.g. fructose found in nectar and fruits to attract insects, and glucose, the main respiratory substrate. Glucose occurs in 2 forms. 2.2 to 1 description and drawing of alpha-glucose and beta-glucose. Glucose, also known as dextrose, is the most common simple sugar, monosaccharide. It has 2 isomeric structures, i.e. dextrose-D, glucose and lactose-L, glucose. The dextrose-D, glucose contains 2 further isomers, i.e. alpha, alpha symbol, and beta, beta symbol, glucose. The key difference between alpha, alpha symbol, and beta, beta symbol, glucose is the orientation of hydroxyl, OH, group attached to the first carbon atom. The two-dimensional graphical representation of these isomeric glucose structures shows that the alpha-glucose has 1 hydroxyl and, for hydroxyl, orientations on the same side. On the other hand, beta-glucose has the orientations of 1 hydroxyl and, for hydroxyl, on opposite sides. By relative stability, alpha and beta-glucoses exist naturally as 36, 64 respectively. Lecture notes for AS Level Biology by Mohammad Rashid. Chat the cell number 03004610455735. The following figure clearly depicts the geometrical orientation of hydroxyl groups on alpha, alpha symbol, glucose and beta, beta symbol, glucose structures. Besides this main difference, i.e. different orientation of hydroxyl group at first carbon atom, the rest of entire structures of alpha, alpha symbol, glucose and beta, beta symbol, glucose are the same. Difference between alpha, alpha symbol and beta, beta symbol, glucose. Similarities among alpha, alpha symbol and beta, beta symbol, glucose. Both alpha, alpha symbol, and beta, beta symbol, glucose structures are simple sugar monomers. They can be crystallized from their aqueous solutions, although alpha, alpha symbol, glucose cannot be crystallized as alpha-glucopyranose hydrate. They both have the same number of chiral carbon centers, i.e. 4. Both alpha, alpha symbol, and beta, beta symbol, glucose are optically active organic compounds. 2.2 to 2 monomers, polymers, macromolecules, monosaccharides, disaccharides and polysaccharides. Biological molecules are based on a small number of chemical elements and frequently consist of monomers joined by condensation into polymers. These polymers may be broken down by hydrolysis to give the monomers from which they are built. Monosaccharides are the basic units, monomers, which make up all other carbohydrates. They are soluble in water, taste sweet and consist of small molecules, characterized by their number of carbon atoms. Polysaccharides are polymers formed by multiple condensation reactions of monosaccharides. They are not sweet or soluble in water and are not crystalline. Their main function in plants and animals is to provide structure and storage because they are insoluble and compact. A macromolecule is a very large molecule important to biophysical processes, such as a protein or nucleic acid. It is composed of thousands of covalently bonded atoms. Many Macromolecules are polymers of smaller molecules called monomers. The term sugars describes monosaccharides and disaccharides. Their names end in a suffix ___ose, e.g. glucose and fructose, etc. Disaccharides, such as maltose, sucrose, cane sugar, and lactose, milk sugar, consist of two monosaccharides linked together. Three common disaccharides are 1. Sucrose, alpha-glucose plus beta-fructose. Transport carbohydrate in plant phloem tissue. 2. Maltose, alpha-glucose plus alpha-glucose. Found in germinating seeds. 3. Lactose, alpha-glucose plus galactose. Found in milk, providing the energy source for suckling mammals. 4. Polysaccharides are polymers formed by multiple condensation reactions of monosaccharides. They are not sweet or soluble in water and are not crystalline. Their main function in plants and animals is to provide structure and storage because they are insoluble and compact. Lecture notes for AS Level Biology by Muhammad Rashid. Chat the cell number 03004610455736. Starch glycogen cellulose polymer of alpha-glucose found in two form, amylose, straight unbranched helix amylopectin, branched chain, branching every 24th glucose monomer. Polymer of branched chains of alpha-glucose, branching every 10th glucose monomer. Polymer of beta-glucose in an unbranched chain. OH groups stick outwards forming H bonds with neighboring chains giving a high tensile strength and permeability. Storage carbohydrate in plants, found in chloroplasts and root cortex parenchyma cells. The large size of the molecules make starch insoluble and can be used for food storage in plants. Storage carbohydrate in animals, found in muscles and liver, readily hydrolyzed to glucose. It consists of alpha-glucose units in more branched and compact chains. It is a mean of food storage in animals due to its compact shape that is not bulky. From plant cell wall, combined with a gel-like organic matrix. The cross bridges give cellulose its tough and resistant properties to provide structural strength to cell walls. 2.2 to 3 Role of covalent bonds in joining similar molecules. Carbohydrates are made up of monosaccharides linked together into polysaccharide chains by a type of covalent bond known as a glycosidic bond. These glycosidic bonds are formed in a dehydration synthesis reaction. The monosaccharides can link together to form a disaccharide. The bond between them is a glycosidic linkage formed in a condensation reaction. Starch and glycogen are formed by the linking of alpha-glucose molecules with the formation of glycosidic bonds. Monosaccharides include glucose, fructose, and galactose. These sugars are isomers. They have the same formula but their atoms are arranged in different ways. Monosaccharides are linked by glycosidic bonds. These are formed by condensation reactions. Two point 2 to 6 Breakage of glycosidic bond by hydrolysis. Glycosidic linkages are hydrolyzed, or broken, through the addition of a water molecule and a catalyst. Carbohydrates bond with each other through glycosidic linkages. These bonds form via a dehydration reaction, also known as a condensation reaction or dehydration synthesis. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455737. 2.2 to 5 glycosidic bond by condensation with reference to Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455738. 2.2 to 7 Molecular structure of polysaccharides, starch, amylose and amylopectin, glycogen. A polysaccharide is a complex carbohydrate polymer formed from the linkage of many monosaccharide monomers. One of the best-known polysaccharides is starch, the main form of energy storage in plants. Glycogen is an even more highly branched polysaccharide of glucose monomers that serves the function of storing energy in animals. Cellulose is another polymer of glucose. It is the structural component of the cell walls of green plants. 2.2 to 8 Molecular structure of cellulose and its role in plant cell walls. It is used in the diet as a fiber supplement. It is used to produce paperboard and paper products. It helps as an additive in various food items. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455739. Glucose monomers. They differ only in the direction that H and OH groups point on carbon 1. When alpha-glucose molecules are joined chemically to form a polymer, starch is formed. When beta-glucose molecules are joined to form a polymer, cellulose is formed. 2.2 to 9 Lipids/fats, triglycerides. Nonpolar hydrophobic molecules. Lipids contain carbon, hydrogen and oxygen, although lower amounts of oxygen than carbohydrates, and are compact and insoluble. The general formula is CnH2nO2. Non-essential fatty acids can be synthesized by the body from carbohydrate and protein metabolism. Essential fatty acids cannot be synthesized and must be obtained from the diet, e.g. vegetables and seed oils. Lipids contain carbon, hydrogen, oxygen, often phosphorus and occasionally nitrogen. Most are nonpolar chemicals and therefore insoluble in water. Lipids are used for energy storage, protection and insulation. Cholesterol is a normal constituent of cell membranes. Storage of metabolic fuels in adipose tissue of animals and oils in plants, e.g. castor oil, seeds and fruits. Lipids contain double the amount of energy of carbohydrates. Heat insulation in hibernating in marine mammals, e.g. whales. Buoyancy and waterproofing in aquatic animals, e.g. geese. February 2nd, 10 Molecular structure of triglycerides and their functions in livings. A triglyceride is composed of one molecule of glycerol and 3 fatty acids. A triglyceride is formed from a condensation reaction called esterification. Fatty acids with double bonds are said to be unsaturated and have a lower melting point than fatty acids with no double bonds. A molecule of glycerol and 3 molecules of fatty acids combine to produce a triglyceride. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455740. February 2nd, 11 Molecular structure of phospholipids. Phospholipids are one of the primary components of the cell membranes. The phospholipid membrane or lipid bilayer is selectively permeable and thus regulates the entry and exit of materials/molecules in the cell. Phospholipids impart cellular membrane fluidity and flexibility. Phospholipids also help in cellular signal transduction. Phospholipids are essential for the availability of polyunsaturated N,3 fatty acids, like docosahexaenic acid, DHA, for the brain and nervous system. Thus, phospholipids are critical for the normal functioning of the nervous system. Cephalin phospholipid is found in the brain, specifically in the white matter of the brain. Dipalmitoyl phosphatidylcholine or DPPC surfactant is the phospholipid found in the lungs. It is a very essential phospholipid that helps in reducing surface tension and preventing alveolar collapse while expiration phenomena. Surfactant phospholipid mixture is also administered to prematurely born newborn babies in order to open up the lungs for respiration. Phospholipids along with cholesterol are important membrane components. The cholesterol-phospholipid ratio controls the immunological functioning of the lymphocytes. With the age, the amount of cholesterol increases, and thus, changes the cholesterol-phospholipid ratio. This affects the immunological functioning of the lymphocytes. High intake of alcohol results in a reduction in the number of phospholipids in the hepatic or liver cells. This leads to reduced flexibility of the hepatic cell membrane, eventually leading to liver injury. Reports suggest that the administration of phospholipids helps to recover from hepatic cell damage. Cell polarity is maintained with the help of the phospholipid binding capacity of polarity regulators of the cell. One of the endogenous phospholipids is plasmalogens. Those functions as antioxidant protecting organs from oxidative stress. These antioxidant phospholipids also help in the significant reduction of oxidized phospholipids in the cell. Phospholipids are essential for lipid metabolism and absorption. Dietary supplementation of phospholipids is reported to inhibit prostaglandin synthesis and thus exert an anti-inflammatory effect. Phospholipids are widely used as a drug delivery vehicle in the pharmaceutical industry. One of the most commonly explored pharmaceutical dosage form for the delivery of anti-cancer drugs is liposomes. Liposomes are primarily made up of phospholipids, example, hydrogenated soy phosphatidylcholine, hydrogenated egg PC or egg phosphatidylcholine, soy lecithin, egg lecithin, etc., and have a unique bilayer spherical structure due to characteristic feature of self-assembly of phospholipids. Further, phospholipids, like egg phosphatidylcholine, soy phosphatidylcholine, and lecithin are widely used as emulsifiers in the pharmaceutical formulation development. Phospholipids are very good drug solubilizers, as they are amphiphilic in nature. Hence, in pharmaceutical phospholipids are one of the primary components of the cell membrane. The phospholipid membrane or lipid bilayer is selectively permeable and thus regulates the entry and exit of material molecules in the cell. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457040. February 2nd, 11 Molecular structure of phospholipids. Phospholipids are one of the primary components of the cell membranes. The phospholipid membrane or lipid bilayer is selectively permeable and thus regulates the entry and exit of materials/molecules in the cell. Phospholipids impart cellular membrane fluidity and flexibility. Phospholipids also help in cellular signal transduction. Phospholipids are essential for the availability of polyunsaturated N,3 fatty acids, like docosahexaenic acid, DHA, for the brain and nervous system. Thus, phospholipids are critical for the normal functioning of the nervous system. Cephalin phospholipid is found in the brain, specifically in the white matter of the brain. Dipalmitoyl phosphatidylcholine or DPPC surfactant is the phospholipid found in the lungs. It is a very essential phospholipid that helps in reducing surface tension and preventing alveolar collapse while expiration phenomena. Surfactant phospholipid mixture is also administered to prematurely born newborn babies in order to open up the lungs for respiration. Phospholipids along with cholesterol are important membrane components. The cholesterol-phospholipid ratio controls the immunological functioning of the lymphocytes. With the age, the amount of cholesterol increases, and thus, changes the cholesterol-phospholipid ratio. This affects the immunological functioning of the lymphocytes. High intake of alcohol results in a reduction in the number of phospholipids in the hepatic or liver cells. This leads to reduced flexibility of the hepatic cell membrane, eventually leading to liver injury. Reports suggest that the administration of phospholipids helps to recover from hepatic cell damage. Cell polarity is maintained with the help of the phospholipid binding capacity of polarity regulators of the cell. One of the endogenous phospholipids is plasmalogens. Those functions as antioxidant protecting organs from oxidative stress. These antioxidant phospholipids also help in the significant reduction of oxidized phospholipids in the cell. Phospholipids are essential for lipid metabolism and absorption. Dietary supplementation of phospholipids is reported to inhibit prostaglandin synthesis and thus exert an anti-inflammatory effect. Phospholipids are widely used as a drug delivery vehicle in the pharmaceutical industry. One of the most commonly explored pharmaceutical dosage form for the delivery of anti-cancer drugs is liposomes. Liposomes are primarily made up of phospholipids, example, hydrogenated soy phosphatidylcholine, hydrogenated egg PC or egg phosphatidylcholine, soy lecithin, egg lecithin, etc., and have a unique bilayer spherical structure due to characteristic feature of self-assembly of phospholipids. Further, phospholipids, like egg phosphatidylcholine, soy phosphatidylcholine, and lecithin are widely used as emulsifiers in the pharmaceutical formulation development. Phospholipids are very good drug solubilizers, as they are amphiphilic in nature. Hence, in the pharmaceutical industry, for solubilization of poorly soluble drugs, phospholipids are used. Phospholipids in plants function in the control of the growth of plants and in the management of stress. In living things there are two main types of lipid, triglycerides and phospholipids. Triglycerides are the familiar fats and oils. Phospholipids form cell membranes. Fatty acids vary in chain length and may be saturated with hydrogen or unsaturated. These factors determine the properties of the triglyceride, such as its melting point and viscosity. Phospholipids are similar to triglycerides, but phosphoric acid replaces one of the fatty acids. They have a polar head, allowing them to form bilayers, membranes, in water. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457041. 2.3 Proteins. Different amino acids can be separated by paper chromatography. A concentrated spot of the mixture of amino acids is placed on the paper and put into chromatography tank. The solvent moves up the paper and the solvent front is marked. The paper is dried and sprayed with ninhydrin to see the position of the amino acids, and their RF values are calculated using the equation. The RF values identify the amino acids. Chromatography can be used to separate and identify molecules in a mixture. Proteins consist of amino acids linked by peptide bonds. There are 20 different amino acids in living things. All have a carboxylic acid group and an amino group but differ in their R group. Amino acids may be linked together with peptide bonds to produce dipeptides and polypeptides. Polypeptides form proteins which show different levels of structure. Interactions between nonpolar R groups of different amino acids form hydrophobic interactions. This causes the protein to fold as hydrophobic side groups are shielded from water. Where R groups contain sulfur, e.g. cysteine, disulfide bonds can be formed between these groups, which contribute to the maintenance of the tertiary protein structure. 2.3 to 1 General structure of an amino acid. Formation and breakage of peptide bond proteins contain carbon, hydrogen and oxygen, along with nitrogen and often sulfur and phosphate. They are polymers of 20 types of amino acids. Amino acids have the following characteristics. Amino acids are amphoteric, acting as both an acid and base. Through condensation of two amino acids they form dipeptides. In chromatography, the amino acids can be identified by calculating the RF value. Amino acids are amphoteric, acting as both an acid and a base. Through condensation. Lecture notes for AS Level Biology by Muhammad Rashid. Chat the cell number 0300. Lecture notes for AS Level Biology by Muhammad Rashid. Chat the cell number 0300-461-045741. 2.3 Proteins. Different amino acids can be separated by paper chromatography. A concentrated spot of the mixture of amino acids is placed on the paper and put into chromatography tank. The solvent moves up the paper and the solvent front is marked. The paper is dried and sprayed with ninhydrin to see the position of the amino acids, and their RF values are calculated using the equation. The RF values identify the amino acids. Chromatography can be used to separate and identify molecules in a mixture. Proteins consist of amino acids linked by peptide bonds. There are 20 different amino acids in living things. All have a carboxylic acid group and an amino group but differ in their R group. Amino acids may be linked together with peptide bonds to produce dipeptides and polypeptides. Polypeptides form proteins which show different levels of structure. Interactions between nonpolar R groups of different amino acids form hydrophobic interactions. This causes the protein to fold as hydrophobic side groups are shielded from water. Where R groups contain sulfur, e.g. cysteine, disulfide bonds can be formed between these groups, which contribute to the maintenance of the tertiary protein structure. 2.3 to 1 General structure of an amino acid. Formation and breakage of peptide bond proteins contain carbon, hydrogen and oxygen, along with nitrogen and often sulfur and phosphate. They are polymers of 20 types of amino acids. Amino acids have the following characteristics. Amino acids are amphoteric, acting as both an acid and base. Through condensation of two amino acids they form dipeptides. In chromatography, the amino acids can be identified by calculating the RF value. Amino acids are amphoteric, acting as both an acid and a base. Through condensation of two amino acids they form dipeptides. The primary structure of a protein is the sequence of its amino acids. Further structures are controlled by the formation of weak hydrogen bonds and disulfide bridges between amino acids. Amphoterics act as both an acid and a base depending on surrounding pH. This allows them to act as a buffer to pH change. Zwitterions. They possess both a negative and positive change. Form dipeptides through condensation of two amino acids. Polypeptides are polymers of amino acids joined by peptide bonds. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610454742. 2.3 to 2 Primary, secondary, tertiary and quaternary structures of proteins. Protein structure. Polypeptides link together to form proteins, which are arranged into 4 levels of protein structure. 1. Primary structure. This structure refers to the sequence of amino acids in a polypeptide. This is a sequence of amino acids determined by the genetic code of DNA. 2. Secondary structure. This structure refers to the twisting of the polypeptide chain into an alpha helix, for example, keratin, or a beta-pleated sheet, for example, fibrin, due to hydrogen bonds between hydrogen and oxygen atoms. They are held together by hydrogen bonds. 3. Tertiary structure. This structure refers to the folding and bending of the polypeptide on itself into a compact globule, for example, all enzymes, due to a combination of hydrogen bonds, ionic bonds, disulfide bonds, and hydrophobic interactions. The secondary structure, one polypeptide chain, is further folded and is held together by hydrogen and disulfide bonds, formed between two cysteine amino acids. These proteins can either be fibrous proteins which have structural functions and are insoluble, e.g. collagen, or globular proteins which are folded as spherical molecules and are soluble, e.g. enzymes. In globular proteins, the hydrophilic amino acids are found on the outside and the hydrophobic amino acids are found on the inside. This affects the folding of the protein. 4. Quaternary structure. This structure refers to the combination of more than one polypeptide, usually associated with non-protein groups, prosthetic groups, for example, in hemoglobin. This structure is due to a combination of hydrogen bonds, ionic bonds, and hydrophobic interactions maintaining the polypeptides together. These proteins consist of 2 or more polypeptide chains, e.g. hemoglobin is composed of 4 polypeptide chains, 2 alpha chains, and 2 beta chains with an attached heme group containing iron. 2.3 to 3 Types of interactions to hold protein molecules in shape: hydrophobic interactions, hydrogen bonding, ionic bonding, covalent bonding and disulfide bond. Types of bonding. 4 types of bonding are involved in the maintenance of protein structure: hydrogen bonds, ionic bonds, hydrophobic interactions, and disulfide bridges. 1. Hydrogen bonds. A hydrogen bond is formed when 2 electronegative atoms, e.g. nitrogen and oxygen, interact with the same hydrogen. These bonds help to contribute to the stability of protein structure. In globular proteins, hydrogen bonding is responsible for the formation of secondary protein structure such as the alpha helix and beta sheet, comprises 80% to 90% of globular protein structure. Formation of hydrogen bonds can occur between. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610454743. Atoms on amino acid and water molecules at the protein surface. Atoms on amino acid and protein backbone atoms. Backbone atoms and water molecules at the protein surface. Backbone atoms on 2 different amino acids. 2. Ionic bonds. Ionic bonds are formed and amino acids carrying opposing electrical charges are next to the hydrophobic core of proteins. As most charged amino acids are found on the protein surface, most ionic bonds occur on the exterior of the protein. Ionic bonds play an important role in protein structure because they provide electrostatic attractions that are almost as strong as covalent bonds. Ionic bonds can also form when a hydrogen ion transfers from the hydroxyl group, COOH, of one amino acid to the amine group, NH2, of another amino acid. 3. Hydrophobic interactions. Hydrophobic bonds are the main driving force for the folding in globular protein structure. They form a hydrophobic core in the interior of the protein, where the hydrophobic side chains are shielded from any interactions with water molecules. The hydrophobic side chains have nonpolar groups that will repel water molecules and other polar groups. 4. Disulfide bridges, SS. The linkage is also called an SS bond or sometimes a disulfide bridge and is usually derived by the coupling of two thiol groups. These disulfide bridges are very strong covalent bonds that help to hold different protein chains or loops within a single polypeptide chain. They also play an important role in holding the catalytic site of an enzyme in its correct configuration to enable the enzyme to function. 2.3 to 4 Globular and fibrous types of proteins. Proteins can either be globular proteins or fibrous proteins. Globular proteins differ from fibrous proteins in 6 ways. Globular proteins. Fibrous proteins. 1. It has polypeptide chains with irregular sequences of amino acids. It has polypeptide chains with regular repetitive sequences of amino acids. 2. Its shape is a compact globule of polypeptides. It has long chains running parallel. 3. It is chemically less stable and its activity is affected by factors such as its concentration, pH and temperature. It is chemically stable and relatively unaffected by concentration, pH and temperature. 4. Each molecule of the same type of globular protein has a specific sequence. Each molecule of the same type of fibrous protein may vary in length with slightly different sequences of amino acids. 5. It is water-soluble. It is insoluble in water. 6. It is involved in various body systems such as the digestive system, the endocrine system and the immune system. Its role is mainly in helping to maintain structure and providing support. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457744. 2.3 to 5 Structure of hemoglobin molecule. An example of a globular protein is hemoglobin. It is also conjugated protein, i.e. having a prosthetic group, and consists of sequences of amino acids forming 4 polypeptide chains that twist and fold into a compact shape and has heme as the prosthetic group. 2 chains are alpha chains and the other 2 are beta chains held together by hydrophobic interactions, hydrogen and ionic bonds. 2.3 to 6 Functions of hemoglobin with reference to iron in the heme group. Hemoglobin has a quaternary structure, with each heme group containing a ferrous iron atom that is able to carry 1 oxygen molecule. Therefore, each human hemoglobin molecule is capable of carrying 4 oxygen molecules, making it an efficient respiratory pigment. This enables it to serve its vital function in oxygen transport. 2.3 to 7 Collagen as fibrous protein. An example of a fibrous protein is collagen. It consists of a primary structure of a repetitive tripeptide sequence that forms long chains and may run parallel to each other. Its secondary structure has 3 unbranched polypeptide chains linked to one another via cross bridges and forming a triple helix. These cross bridges involve strong covalent bonds that help provide structural support to the collagen molecule, thereby making it resistant to stretching. Hence, collagen has an important structural role in providing physical strength to connective tissue such as tendons. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610457745. 2.3 to 8 Structure of collagen molecules and collagen fibers and their function. Collagen is composed of 3 chains. The chains are wound together to form a triple helix. Since glycine is the smallest of all the amino acids, it allows the chain to form a tight configuration, and it can withstand stress, helping fibroblasts to form in your dermis, middle skin layer, which helps new cells grow, playing a role in replacing dead skin cells, providing a protective covering for organs, giving structure, strength and elasticity to your skin, helping your blood to clot. 2.4 Water. Water molecules have 2 charged ends and can form weak bonds. Water is said to be a dipolar molecule. Water is a dipolar molecule with 2 charged ends, which attract ions and other charged molecules, forming weak hydrogen bonds. This makes water an excellent solvent, as water molecules form shells around the solute molecules, e.g. Na+. It also allows cohesion, water molecules cling together, and adhesion, water molecules stick to other polar molecules. Water has the following properties. Property significance in living system. The polar nature of water. This makes water a very good solvent. It has been called the universal solvent. Though obviously not true, since it doesn't dissolve everything, it does act as a solvent for all ionic and polar compounds. High specific heat capacity. A gram of water absorbs a lot, 4.2 joules, of heat before its temperature rises by 1 OC. Organisms have high water content and so can absorb a lot of energy, or lose a lot, before their temperature changes significantly. This makes water a useful thermal buffer. Most organisms cannot tolerate great changes in temperature, and the water in their bodies protects them from rapid changes in external temperature. High latent heat of vaporization. The hydrogen bonding between water molecules makes it difficult for them to separate from each other and vaporize, evaporate, to form a gas. When water does evaporate, the escaping molecules take a lot of energy with them, and so vaporization is a very efficient cooling process. Anyone who has ever stepped out of the shower and stood in a draft will know just how efficient. The process of sweating, panting and transpiration all involve cooling by evaporation. A large amount of heat energy is needed to turn a given amount of liquid water into water vapor. This is used in sweating to cool down the body temperature. Lecture notes for AS Level Biology by Muhammad Rashid. Chat the cell number 03004610454746. High latent heat of fusion. A large amount of heat needs to be lost before water freezes. Cell contents are less likely to be damaged by ice crystals. Small relative molecular mass, 18. It is liquid at room temperature, needed for chemical reactions. Melting and boiling point. Water freezes at 0 OC and is therefore a liquid over most of the temperature range found on Earth. Thermal conductivity. A naked person sitting in a room at 25 OC would find it comfortable. However, if immersed in water at 25 OC, the same person would feel cold. This is because the thermal conductivity of water allows heat to be transferred away from the body more quickly through the water than through air. Materials with a low thermal conductivity are good insulators. Fur is a good insulator because it traps air, but the insulating properties are lost if the fur gets wet. High specific heat capacity. A large amount of heat energy is needed to raise the temperature of 1 G of water by 1 OC. This maintains a constant internal environment where temperature changes are minimized in cells. Ice formation. Ice is less dense than water and floats, which insulates the water below and allows aquatic life to survive. High surface tension. High surface tension allows movement of small insects. Hydrolysis. Water is used to split up large organic molecules into monomers. Cohesion. Hydrogen bonding causes water molecules to stick to each other. This is important, for example, When water evaporates from the leaves of a tall tree, water is so cohesive that a continuous column can be sucked up from roots which might be over 100 meters below. This cohesion causes surface tension. Water has a very high surface tension, and several organisms, such as pond skaters and rat-tailed maggots, take advantage of this property. Key words: biological molecules, biochemistry, the building blocks of life, alpha-glucose, amino acids, amylopectin, amylose, Benedict's test, beta-glucose, biuric test, cellulose, collagen, condensation reaction, disaccharide, disulfide bond, emulsion test, fibrous proteins, globular proteins, glycogen, glycosidic bond, hemoglobin, hydrogen bond, hydrolysis reaction, hydrophobic interaction, iodine test, latent heat of vaporization, macromolecule, monomers. Monosaccharide, non-reducing sugar, peptide bond, phospholipid, polymers, polysaccharide, primary structure, quaternary structure, reducing sugar, secondary structure, solvent, specific heat capacity, starch, sucrose, tertiary structure, triglyceride. Chapter Glossary. Alpha-glucose is a structural isomer of glucose that exists in a ring form where the hydroxyl group on carbon-1 lies below the plane of the ring. Beta-glucose is a structural isomer of glucose that exists in a ring form where the hydroxyl group on carbon-1 lies above the plane of the ring. Amino acids are the monomers containing an amino group, NH2, a carboxyl group, COOH, and a variable R group that make up proteins. Amylopectin is a branched polysaccharide made up of alpha-glucose monomers joined by alpha-1,6 glycosidic bonds that makes up starch along with amylose. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610454747. Amylose is an unbranched polysaccharide made up of alpha-glucose monomers joined by alpha-1,4 glycosidic bonds that makes up starch along with amylopectin. Benedict's test is a biochemical test for reducing sugars that produces a different color based on the amount of reducing sugar present. Biuric test is a biochemical test that produces a purple color when it is added to a solution containing protein. Cellulose is a polysaccharide made of beta-glucose monomers joined by beta-1,4 glycosidic bonds that is used as a structural polysaccharide, providing strength to plant cell walls. Collagen is a type of fibrous protein that provides strength to many different cell types and makes up connective tissues. Condensation reaction is a type of reaction that joins two molecules together with the formation of a chemical bond involving the elimination of a molecule of water. Disaccharide are molecules formed by the condensation of two monosaccharides. Disulfide bond is a covalent bond formed between sulfur-containing cysteine side chains of amino acids. Emulsion test is a biochemical test that produces a cloudy emulsion when performed on lipids. Fibrous proteins is a class of long-chain proteins that are generally insoluble in water and typically have structural roles. Globular proteins is a class of spherical-shaped proteins that are generally water-soluble and typically have metabolic roles. Glycogen is a highly branched polysaccharide made of alpha-glucose monomers that is used as the main storage of energy in humans and animals. Glycosidic bond is a bond between two monosaccharides formed in a condensation reaction. Hemoglobin is a type of conjugated globular protein used to transport oxygen that is made up of 4 polypeptide chains, each containing a heme prosthetic group. Hydrogen bond is a type of weak bond formed between an electropositive hydrogen and an electronegative atom like oxygen or nitrogen. Hydrolysis reaction is a type of reaction involving the use of a water molecule that breaks a chemical bond between two molecules. Hydrophobic interaction. Polar side chains face the cytosol. Nonpolar side chains arrange to minimize the surface area in contact with water and often point inwards. Iodine test is a biochemical test that produces a blue/black color when it is added to a solution containing starch. Latent heat of vaporization is the amount of energy needed for a substance to change state from a liquid to a gas. Macromolecule is a large molecule formed by condensation reactions between smaller molecules. Polymers are a type of macromolecule, but not all macromolecules are formed from repeating units. Monomers are the smaller units from which larger molecules are made. Monosaccharide are the individual sugar monomers from which larger carbohydrates are made. Non-reducing sugar is a sugar that does not have a free aldehyde or ketone functional group, so cannot act as a reducing agent, e.g. sucrose. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610454748. Peptide bond is the bond between amino acids formed by a condensation reaction between the H of the amine group on one molecule and the OH of the carboxylic acid group on the other molecule. Phospholipid is a type of lipid formed by the condensation of one molecule of glycerol, two molecules of fatty acid and a phosphate group. The two fatty acid chains are the non-polar hydrophobic tails, whilst the phosphate group is the polar hydrophilic head. Polymers are molecules made from a large number of monomers joined together. Polysaccharides are molecules formed by the condensation of many monosaccharides. Primary structure is the individual sequence of amino acids in a protein. Quaternary structure is a structure only applicable to proteins with multiple polypeptide chains that describes the interactions of the different chains. Reducing sugar is a sugar that has a free aldehyde or ketone functional group, so can act as a reducing agent, e.g. maltose, fructose, glucose. Secondary structures are the local interactions of the amino acids in the polypeptide chain resulting in an alpha helix or a beta-pleated sheet. Solvent is a substance in which other solutes are dissolved. Specific heat capacity is the amount of energy needed to raise the temperature of a substance by a specific amount. Starch is a polysaccharide made of alpha-glucose monomers that is used as the main storage of energy in plants. Sucrose is a disaccharide formed by condensation of a glucose molecule and a fructose molecule. Tertiary structure is the way that the whole protein folds to make a three-dimensional structure. Triglyceride is a type of lipid formed by the condensation of one molecule of glycerol and three molecules of fatty acid. Short MCQs with right answers. Carbohydrates. 1. The table shows some steps that can be made in carrying out the Benedict's test. 2. Which combination of steps is required to carry out a semi-quantitative test on a reducing sugar solution? 3. The diagrams show three examples of different bonds. Which bonds hold the secondary structure of proteins together? One only. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610454749. 4. Insulin is a globular protein involved in cell signaling. It is transported in the blood plasma from the cells that synthesize it to its target cells. A molecule of insulin contains 6 sulfur-containing amino acids and has 2 polypeptide chains. Which statements about insulin are correct? 1. An insulin molecule has a quaternary structure. 2. Amino acids with hydrophobic R groups would be found in the center of an insulin molecule. 5. Which statement correctly explains why blood plasma can be maintained at a stable temperature? It has a high specific heat capacity. 6. Which set of steps is the best method for conducting the emulsion test for lipids? Add 2 cubic centimeters of ethanol to the sample and shake. Pour the ethanol into a test tube containing 2 cubic centimeters of water and shake again. Lipids are present if the mixture becomes cloudy. 7. A student was provided with a solution of carbohydrate. They removed 2 samples from the solution and perform tests on each sample, as shown. 8. Which statement explains the results? Glycosidic bonds in a polysaccharide have been broken to release reducing sugar. 9. Which molecules contain at least 2 double bonds? Collagen and hemoglobin, option B. 10. 11. What describes cellulose? An unreactive linear chain of 1 to 4 beta-glucose 12. Which part of the structure of hemoglobin carries oxygen? Heme groups. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455750. 13. What is the maximum number of hydrogen bonds that can form between two single water molecules? 114. How is non-reducing sugar tested? Add a few drops of acid, then heat, neutralize with alkali, add 6 cubic centimeters of Benedict solution and heat it again. 15. Which features contribute to the function of a cellulose molecule? 1. Many hydrogen bonds form between adjacent chains. 2. It is insoluble in water. 16. What correctly describes triglycerides? Nonpolar molecules that are soluble in ethanol. 17. Cocoa butter contains 3 different triglycerides. These triglycerides are made from the fatty acids oleic acid, O, palmitic acid, P, stearic acid, S. 18. The 3 triglycerides found in cocoa butter are POS, SOS and POP. The chemical structure of the triglyceride POS is shown next to a diagrammatic representation of POS. 19. Which statement is correct? Triglyceride SOS contains 4 more carbon atoms than triglyceride POP. 20. Which molecules contain at least 4 double bonds? Unsaturated triglyceride, hemoglobin, and collagen, option D. 21. 22. The diagram shows the amino acid glutamic acid. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455751. 23. 24. What is the R group for glutamic acid? CH2CH2COOH25. The diagram shows 3 interactions that hold protein molecules in shape. 26. Which row identifies these interactions? 27. Dialysis, Vis-King, tubing is an artificial partially permeable membrane with pore sizes of approximately 2.5 nanometers. Glucose molecules have a diameter of about 1.5 nanometers and can pass through the pores in the membrane. What else can pass through the pores? Fructose 28. What is present in all viruses? Adenine 29. To estimate the concentration of glucose in an unknown solution, equal volumes of a range of known concentrations of glucose were each mixed with the same excess volume of Benedict solution. After mixing, the solutions were placed in a thermostatically controlled water bath at 90 OC for 3 minutes. The unknown solution was then treated in the same way and the colors of the known and unknown solutions compared. What is the independent variable in this procedure? Concentration of glucose 30. Which statements are correct for amylose and also for amylopectin? 1. They are carbohydrate molecules. 2. They are formed by condensation reactions. 3. They contain 1,4 glycosidic bonds. 31. Which statement is a correct comparison between saturated triglyceride molecules and unsaturated triglyceride molecules of approximately the same molecular masses? Unsaturated triglycerides have more double bonds and fewer hydrogen atoms than saturated triglycerides. 32. Which molecules contain at least 3 double bonds? Saturated triglyceride, collagen and hemoglobin. 33. Which diagram correctly shows hydrogen bonding between 2 water molecules? 34, 35. The diagram represents a molecule from a cell surface membrane. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455752. 36, 37. Which description of one of the labels is correct? Phosphate group at the hydrophilic end of the molecule. 38. Which row correctly shows levels of protein structure that can be held together by each type of interaction? 39. A student used Benedict's solution to test a sample known to contain carbohydrate. At the end of the test the solution was blue. Which carbohydrate could be present in the sample? Sucrose 40. A student carries out a semi-quantitative test with Benedict's solution. Which statement about this procedure is correct? It provides an indication of relative reducing sugar concentration. 41. A sample of food was heated with Benedict's solution which changed color to green. A second sample of the same food was boiled with dilute hydrochloric acid and neutralized using sodium hydrogen carbonate. It was then heated with Benedict's solution which changed color to red. What did these results show? Reducing sugar and non-reducing sugar present. 42. A student carried out the Benedict's test on a sample and got a negative result. What should the student do to confirm that there are no sugars present in the sample? Boil. With HCl, neutralize with alkali and repeat the Benedict's test. 43. Which color indicates the lowest concentration of reducing sugar present in a solution after testing with Benedict's solution? Green. 44. After boiling a sample of milk with Benedict's solution, a yellow color is observed. Which conclusion about the sample of milk is correct? Reducing sugars are present. 45. A sample of solution was tested for reducing sugar and the result was negative. Another sample of the same solution was tested for non-reducing sugar and the result was positive. Which step in the test for non-reducing sugar breaks the glycosidic bonds? Boiling with HCl. 46. A glycosidic bond is broken and two monosaccharides are formed during a positive test for a non-reducing sugar. Which row indicates the catalyst and reactants in the process? Catalyst, HCl, reactants, sucrose and water. 47. Which concentrations could be produced by a serial dilution of an 8% glucose solution? 4%, 2%, 1%, 0.5% and 0.025%. 48. Which carbohydrate gives a brick red color when boiled with Benedict's solution? Fructose. 49. Heating with which solution breaks glycosidic bonds? Dilute HCl. 50. Tests were performed on samples from a mixture of biological molecules. When iodine in potassium iodide solution was added to a sample, the mixture turned black. When the biuret test was carried out on another sample, the mixture turned purple. Which biological molecules were in the mixture? Amylase and starch. 51. A student carried out 4 tests for biological molecules on a solution. The results are shown in the table. Test for biological molecules. Observation: iodine solution orange-brown. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455753. Biuret purple, Benedict's orange, emulsion clear. Which 3 molecules may be present in this solution? Globin, glucose and collagen. 52. A student carried out 4 tests for biological molecules on a solution. The results are shown in the table. Test for biological molecules. Observation: iodine solution orange-brown, biuret purple, Benedict's orange, emulsion cloudy. Which 3 molecules may be present in this solution? Lipid, protein and glucose. 53. Trehalose is the sugar that gives a negative result when tested with Benedict's solution. A molecule of trehalose forms 2 alpha glucose molecules when it is hydrolyzed. Which row is correct? Formula of trehalose, C12H22O11 sugar that gives the same result with Benedict's as trehalose, sucrose 54. Which molecules contain 1.4 glycosidic bonds? Amylose, cellulose and glycogen 55. Complete hydrolysis of polysaccharides requires all the glycosidic bonds between the monomers to be broken. Enzyme X only breaks alpha-1 for glycosidic bonds. Which row shows how completely enzyme X can hydrolyze molecules of glycogen and amylose? Glycogen, some hydrolysis, amylose, most hydrolysis 56. Which statement can be used to describe the structure of cellulose? 1. A polymer of glucose monomers linked by beta-1 for glycosidic bonds 2. A polysaccharide of hexose monomers 3. An unbranched molecule made of beta-glucose monomers 57. Which general formula is correct for monosaccharides such as fructose? CH2O, N58. One molecule of X is formed by a single condensation reaction releasing one molecule of water. What is molecule X? A disaccharide 59. A table shows some information about carbohydrate polymers. Which row describes glycogen? Alpha-1 for glycosidic bond, alpha-1, 6-glycosidic bond, branched molecule used for storage in animals 60. Which row is correct for carbohydrates? Macromolecule is starch having monomers alpha-glucose and the polymer is glycogen. 61. Which polysaccharides contain 1, 4 and 1, 6 glycosidic bonds? Amylopectin and glycogen. 62. Which molecules contain at least 2 double bonds? Unsaturated fatty acid, collagen. And hemoglobin 63. Maltose and sucrose are disaccharides. Maltose is formed from 2 molecules of glucose, whilst sucrose is formed from fructose and glucose. Which row shows the molecular formulae of the 2 disaccharides? Maltose, C12H22O11, sucrose, C12H22O1164. Which statement about biological molecules is correct? Amylopectin, amylose, and cellulose are all polymers. 65. What occurs during the formation of a glycosidic bond between 2 alpha-glucose molecules? A molecule of water is always formed. 66. Which molecules are monomers? Ribose, glucose, and deoxyribose 67. What can occur as result of condensation reaction? A disaccharide is formed, a molecule of water is produced and 2 monosaccharides join together. 68. Which of the statements about polysaccharides can be used to describe amylose and cellulose? 1. Contains 1, 4 glycosidic bonds 2. Polymer of glucose 69. What can occur during condensation of carbohydrate? A disaccharide is produced from monosaccharide. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455754. 70. Which words correctly complete the description of amylose? The polysaccharide amylose is a polymer made up of monomers of the monosaccharide glucose. 71. What is the general formula for glycogen? C6H10O5, N72. Which row correctly identifies 3 polysaccharides? Alpha-glucose straight chain, amylose, alpha-glucose branching chain, amylopectin and beta-glucose straight chain, cellulose 73. Which comparison of bacteria cell walls and plant cell walls is correct? Bacteria cell wall is made up of peptidoglycan and plant cell wall is made up of polymer of beta-glucose. 74. What is the general formula for cellulose? C6H10O5, N75. A solution containing equal masses of amylose and amylopectin are completely hydrolyzed. Which molecules will be found after the hydrolysis? Alpha-glucose only 76. What is the general formula for amylose? C6H10O5, N77. Which bonds will be broken when a molecule of glycogen is broken down? Alpha-1, 4 and alpha-1, 6 only. 78. Which features adapt a cellulose molecule for its function? Many hydrogen bonds form between adjacent chains and it is insoluble in water. 79. Which two polysaccharides both have 1,6-glycosidic bonds and are branched? Amylopectin and glycogen. 80. Which statements about both amylose and amylopectin are correct? They are polymers having alpha-1 for glycosidic bonds. 81. Which statement describes how the molecular structure of starch is suited to its function? The amylose-amylopectin complex is insoluble and does not affect the water potential of the cell. 82. What is the general formula for amylopectin? C6H10O5, N83. Which row about alpha-glucose and beta-glucose molecules is correct? Carbon atom on which the OH position is different. 1. Cellulose contains both molecules. Number 84. Which row describes amylose? Alpha-1 for glycosidic bonds, present, alpha-1, 6 glycosidic bonds, absent, the shape of molecule, helical. 85. What cannot occur as a result of condensation reaction? Breaking of glycosidic bond. 86. What may take place during a hydrolysis reaction? A glycosidic bond is broken and a sucrose molecule is split into fructose and glucose. 87. Which diagram shows the bond linking the individual units of both cellulose and glycogen? C88. Which bonds hold together the structure of cellulose? Glycosidic and hydrogen bonds. 89. Which is the correct description for the structure of amylose and cellulose? Amylose, alpha-glucose, no rotation, 1 for linkages cellulose, beta-glucose, 180 O rotation, 1 for linkages 90. Which diagram shows the bond linking the individual units of both glycogen and amylose? C91. Which bonds will be broken when a molecule of amylose is hydrolyzed? Alpha-1 for glycosidic bonds. Only 92. Which row is correct and one molecule of sucrose is hydrolyzed? Molecules of reducing sugar, 2 and molecules of water, 1, 93. A naturally occurring polysaccharide is a branched chain of alpha-glucose. The straight parts of the molecule are linked by alpha-1, 6 glycosidic bonds with the small number of branches which are linked by alpha-1, 3 glycosidic bond or an alpha-1 for glycosidic bonds. Which polysaccharide has a structure most similar to that described? Amylopectin. Lecture Notes for AS Level Biology by Mohammad Rashid. Chat the cell number 03004610455755. 94. One naturally occurring polysaccharide is an unbranched chain of the molecule acetylglucosamine linked by beta-1 for glycosidic bonds. There are CH2OH groups that alternate on each side of the polysaccharide chains. Which polysaccharide has a structure most similar to that described? Cellulose. 95. What occurs when sucrose is broken down to monosaccharides? Hydrolysis, releasing reducing sugars and using water. 96. Glycogen is a polymer of glucose. Which description summarizes its structure? Alpha-glucose joined by glycosidic bonds involving carbons 1, 4, and 6. 97. Which molecule has a structural formula that contains C=O bonds? Fatty acids and glucose. Only 98. Which statement about polysaccharide can be used to describe both amylopectin and cellulose? Contains 1, 4 glycosidic bonds. Lipids. 99. Long chain, saturated fatty acids change from solid to liquid at higher temperatures compared with short chain unsaturated fatty acids. Which fatty acids would be more likely to form triglycerides in mammals that live in cold climates? Short-chain unsaturated 100. Some foods contain hydrogenated vegetable fats. These are unsaturated fats that have converted to saturated fats. Which property of the fats will have changed? Their hydrocarbon chains will fit together more closely. 101. Which statements about carbohydrates and triglycerides are correct? 1. They contain carbon, hydrogen, and oxygen. 2. They are used as energy stores. 102. Which statement is correct for triglycerides and phospholipids? Hydrocarbon chains may be saturated or unsaturated. 103. Which row correctly completes the sentence? A triglyceride has ester bonds formed by joining fatty acids to glycerol. 104. Which row correctly shows the ratio of carbon atoms to hydrogen atoms for glucose? A saturated fatty acid and sucrose. Glucose, 1:2, saturated fatty acid, 1:2, sucrose. 1 minute and 1.8 seconds 105. Which row shows a correct comparison between a phospholipid molecule and a triglyceride molecule? Phospholipid molecule, fewer ester bonds, triglyceride molecule, more ester bonds 106. Which molecules are formed by condensation which involves a carboxyl group? Collagen, beta-globin, triglyceride 107. What is the maximum number of water molecules produced when a triglyceride is synthesized? 108. Which statements about phospholipids and triglycerides are correct? 1. They both contain ester bonds. 2. They both may have saturated and unsaturated fatty acid chains. 109. What is the maximum number of condensation reactions that occur when a triglyceride is synthesized? 110. Which features of triglycerides make them suitable energy stores? 1. Contains a high proportion of carbon-hydrogen bonds. 2. Insoluble in water 111. Which row describes a triglyceride? Hydrophilic, not correct. Insoluble in alcohol, not correct. Lecture notes for AS Level Biology by Mohammad Rashid. Chat the cell number 03004610455756. 112. What is true about triglycerides? Hydrophobic, correct. Insoluble in alcohol, not correct. 113. Which pair of statements is correct? Glycolipids are found in all cell surface membrane. Carbohydrates are stored as starch in plants. 114. Which feature of phospholipids enables a bilayer to form? They have hydrophobic and hydrophilic components. 115. Phospholipids and triglycerides are important biological molecules. Which properties are correct for these molecules? Nonpolar, triglyceride, partially hydrophobic, phospholipid 116. In unsaturated lipid molecules, where are double bonds located? Within fatty acids only 117. What are the features of triglycerides? Polar, NO, less dense than water, yes, lower proportion of hydrogen than in carbohydrates, number 118. The structure of phospholipids and triglycerides include the following. Which structures enable the formation of a lipid bilayer in cell surface membranes? Hydrophobic fatty acid chains. 119. Which statements about a triglyceride are correct? It is formed using ester bonds. It does not form hydrogen bonds. 120. Which statements about fatty acids are correct? Form part of cell membrane, form storage droplets. 121. Which describes a triglyceride? It is hydrophobic and not insoluble in alcohol. 122. Which describes a triglyceride? Non-hydrophilic and not insoluble in alcohol. 123. In all lipid molecules where are double bonds located? Within fatty acids only 124. Which molecules contain a carboxyl group? Amino acid, saturated fatty acid and unsaturated fatty acid only 125. Which statement about triglyceride is correct? They have a lower ratio of oxygen to carbon compared with carbohydrates. Proteins. 126. Typical enzymes are large globular proteins with a specific tertiary shape. Which molecular interactions are directly involved in maintaining the tertiary shape? Hydrogen bonding, disulfide bridges and hydrophobic interactions 127. A polypeptide contains a specific number of amino acids, N. How many peptide bonds are present in this polypeptide? N, 128. Which row matches each molecule to a type of bond that is present? Ester bond, phospholipid, hydrogen bond, cellulose and disulfide bond, antibody 129. Which row is correct about the structure of protein? Primary, determined by the sequence of amino acids. Secondary, occurs by hydrogen bonding between NH and C equals O groups of amino acids. Tertiary, disulfide, ionic, hydrogen bonds and hydrophobic interactions all are involved in quaternary, always formed by association of two or more polypeptides 130. Which molecule contains at least two double bonds? Hemoglobin and collagen only. 131. Which feature of collagen enables it to fulfill a structural role in skin and in tendons? Collagen molecules are formed as a triple helix of polypeptide chains. 132. Which molecules contain at least three double bonds? Hemoglobin and collagen. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455757. 133. Which molecule contains the smallest number of hydrogen atoms? Glycine, an amino acid in which the R group is H. 134. Which statements about a peptide bond are correct? It contains 4 different atoms and is important in the primary structure of proteins. 135. In a healthy human, the mean value for the number of hemoglobin molecules in one red blood cell is 260 million. How many alpha-globin chains does one red blood cell contain in a healthy human? 5.2 by 108 136. Which description of collagen is correct? A collagen molecule consists of 3 polypeptide chains in which every third amino acid is glycine. The 3 polypeptides are wound tightly together into a triple helix. Many of these helixes form a fiber. 137. Which statement about collage is correct? 3 helices are held together by hydrogen bonds. 138. Lysozyme occurs in human tears, saliva, milk and mucus. Lysozyme has a role in the immune system and hydrolysis peptidoglycan. Which description of lysozyme is correct? It is an extracellular antibacterial protein. 139. Which row correctly identifies structural features of collagen molecule and a hemoglobin molecule? Molecule consists of more than one polypeptide, collagen and hemoglobin, globular protein, hemoglobin only 140. The enzyme trypsin hydrolysis protein to amino acids. Trypsin does not function when the pH is very low, as its 3D shape would be changed. What explains this change in 3D shape? Hydrogen ions attach themselves to negatively charged R groups. 141. What occurs during the formation of a peptide bond between amino acids? OH is removed from COOH and H is removed from NH2. 142. Which type's bond will keep a folded protein in its precise shape for the longest time as the temperature rises? Disulfide bond 143. What is the minimum number of carbon atoms in an amino acid? 144. Which statements about glycosidic bonds are correct? 1. They occur by Condensation reaction between the OH groups of two molecules. 2. Hydrolysis of the bonds releases energy. 145. Which molecules contain the following bonds? Ester, phospholipid, hydrogen, cellulose, disulfide, antibodies. 146. Which molecules are globular proteins? Amylase, hemoglobin and DNA polymerase. 147. When hydrolyzed, which molecules have products containing a carboxyl group? Phospholipids and proteins. 148. A person with diabetes is unable to make enough of the protein insulin. Some forms of diabetes can be treated by using insulin produced by animals. Scientists have compared the amino acids in insulin produced by animals to human insulin. Which level of protein structure were the scientists studying? Primary structure. 149. Which levels of protein structure are maintained by disulfide bonds? Tertiary and quaternary. Only 150. A polypeptide has number of amino acids, n. How many peptide bonds and are groups, side chains, does this polypeptide have? N. 1 peptide bonds and n are groups. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455758. 151. Lysozyme is an enzyme found in tears and saliva. Which statement refers only to the tertiary structure of lysozyme? The 35th and 52nd amino acid helps to form the active site. 152. What describes the primary structure of a protein? The specific order of amino acid. 153. Boiling the bones and teeth from dead animals can be used to produce a type of glue. The glue is formed from the collagen fibers present in the bones and teeth. Which statement describes the changes to collagen that occur when the glue is produced? The helices of collagen molecules unwind. 154. Hemoglobin consists of 2 alpha chains and 2 beta chains. Approximately 5% of all humans have one amino acid in the beta chain that has been changed, affecting the structure and stability of hemoglobin. Which of the level of protein structure could be changed in the hemoglobin of these humans? Primary, secondary, tertiary and quaternary. 155. Some of the molecules found in animal tissues are grouped into lists. Which lists? Include one or more molecules that always contain nitrogen atoms. 1. Glycogen, antibodies, adenine, phospholipids. 2. Hemoglobin, carbon dioxide, mRNA, monosaccharides. 156. Which feature explains why hemoglobin is soluble? The hydrophilic R groups are arranged around the outside of the molecule. 157. What could describe the tertiary structure of a protein? A globular structure and a specific 3D shape. 158. Which statement about the quaternary structure of proteins is correct? Depends on the primary structure of the subunits. 159. Which biological molecules always contain the element nitrogen? Enzymes, mRNA and tRNA. 160. High concentration of urea breaks all bonds except covalent bonds in protein molecules. Which level of protein structure would remain unchanged when a protein is treated with urea? Primary. 161. Which type of bond does not hold together the tertiary structure of a protein? Peptide. 162. In a ribosome, which bond holds together two adjacent amino acids? Peptide. 163. What describes only the quaternary structure of hemoglobin? For polypeptide chains joined together. 164. Which type of bond holds together an alpha helix or beta pleated sheet of a protein? Hydrogen bonds. 165. How many molecules of oxygen are bound to one molecule of hemoglobin when it is fully saturated with oxygen? 166. Which level of organization of a hemoglobin molecule is coded by a DNA molecule? Primary structure 167. Which explains why hemoglobin is able to carry oxygen. It contains iron. 168. The formation of a glycosidic and peptide bonds is responsible for polymerization in some biological molecules. Which bonds are found in which molecules? Glycosidic, glycogen, peptide, glycoprotein 169. Which of the bonds will be last to break as the temperature of an enzyme is increased? Covalent 170. Which substances contain carbon, hydrogen, oxygen and nitrogen? Collagen and deoxyribonucleic acid. 171. Which level of protein structure maintains the globular shapes of enzyme? Tertiary. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455759. Water. 172. Which properties of water reduce temperature changes inside cells? Specific heat capacity 173. Which molecules have properties that are dependent on hydrogen bonds? Cellulose, hemoglobin and water 174. Which properties of water are the results of only cohesion? Water has high surface tension. 175. The statements are about the properties of water. What allows a small insect to rest on the surface of a pond? Ability to form hydrogen bonds with other molecules 176. Polar molecules form hydrogen bonds with each other. Which properties of water result from its molecules being polar? Good solvent, high specific heat capacity, high surface tension and cohesive 177. Which molecule has properties that are not dependent on hydrogen bonds? Glycogen 178. Which feature of water is least likely to affect the life of an animal in a tropical rainforest? Maximum density at 40 Celsius 179. Which of the statements about polysaccharides can be used to describe both? Glycogen and amylopectin. Contain 1 4-glycosidic bond, contain 1,6-glycosidic. Bonds and polymer of alpha-glucose 180. Which row shows the types of bond found at different levels of protein structures? Tertiary structure have hydrogen bond, ionic bond and hydrostatic bond 181. Which molecules have a structural formula that contains C=O bonds? Glucose, proteins, fatty acids and glycerol 182. Which of the statements about polysaccharides can be used to describe both amylose and glycogen? Contain 1 4-glycosidic bond and polymer of alpha-glucose 183. Heating with which solution breaks glycosidic bond? Dilute hydrochloric acid 184. Which molecule contains C=O bonds? Amino acids, proteins and fatty acids 185. Which statement about triglycerides is correct? They have a lower ratio of oxygen to carbon compared with carbohydrates. 186. Which of the bonds will be last to break as the temperature of an enzyme is increased? Covalent 187. Which row is correct for each of the molecules? Beta-glucose, in its ring structure. The hydroxyl group of carbon atom 1 is above the plane of the ring, collagen. Polysaccharide chains interact to produce a fibrous protein, hemoglobin, has all 4 levels of protein structures and at least 4 types of bond, sucrose, digestion yields glucose-fructose in equal proportions 188. Which carbohydrate gives a brick red color when heated with Benedict's solution? Fructose 189. Glycogen is a polymer of glucose. Which description summarizes its structure? Alpha-glucose joined by glycosidic bonds involving carbons 1, 4 and 6. 190. Which substances contain carbon, hydrogen, oxygen and nitrogen? Collagen and deoxyribonucleic acid 191. Which level of protein structure maintains the globular shapes of enzymes? Tertiary 192. Which of the statements about polysaccharides can be used to describe both amylopectin and cellulose? Contains 1,4 glycosidic bonds. Lecture notes for AS Level Biology by Mohammad Rashid. Chat the cell number 03004610455760. 193. Two disaccharides are cellobiose and sucrose. Cellobiose is formed from two molecules of glucose, whilst sucrose is formed from fructose and glucose. Which row shows the molecular formulae of the two disaccharides? Cellobiose, C12H22O11, sucrose, C12H22O11-194. High concentrations of urea break all bonds except covalent bonds in protein molecules. Which level of protein structure would remain unchanged when a protein is treated with urea? Primary 195. Which property of water makes it most suitable for transport in eukaryotic organisms? Solvent properties 196. Two disaccharides are maltose and sucrose. Maltose is formed from two molecules of glucose, whilst sucrose is formed from fructose and glucose. Which row shows the molecular formulae of the two disaccharides? Maltose, C12H22O11, sucrose, C12H22O11-197. Which row describes a triglyceride? Non-hydrophilic and soluble in alcohol. 198. Myoglobin is a protein with a similar function to hemoglobin. However, myoglobin does not have a quaternary structure. Why does myoglobin not have a quaternary structure? Myoglobin has only one polypeptide chain. 199. In which levels of protein structure do ionic bonds occur? Tertiary and quaternary 200. Which polysaccharides are branched and which are unbranched? Branched, amylopectin; unbranched, cellulose 201. Some foods contain hydrogenated vegetable oils. These are unsaturated fats that have been converted to saturated fats. Which property of the fats will have been changed? Their hydrocarbon chains will fit together more closely. 202. Which set of statements correctly describes hemoglobin? For polypeptide chains, each containing a prosthetic group, iron ions can associate with oxygen forming oxyhemoglobin. In each chain, hydrophobic arc groups of amino acids point towards the center of the molecule. At 50% saturation, two oxygen molecules are transported by the molecule. 203. Which molecular bonds will be broken by hydrolysis when a molecule of glycogen is converted to glucose? 1,4 bonds and 1,6 bonds of alpha-glucose 204. Which juice contains the greatest mass of non-reducing sugar? Mass of precipitate before hydrolysis/mg, 30, mass of precipitate after hydrolysis/mg, 55-205. Water is important for living organisms because it acts as excellent solvent for transport of dissolved glucose and urea. Which linkages are found between the glucose units in cellulose? Beta 1 to 4 only 206. A peptide bond is formed between atoms of which two elements? Carbon and nitrogen 207. An amino acid can be represented as where A represents a variable side chain, which is not a possible side chain. HOC2CH, CH2O208. Which statements about amylopectin and glycogen are correct? Both contain 1 to 4 glycosidic bonds. Glycogen contains more 1 to 6 branches than amylopectin 209. Which feature distinguishes starch from glycogen? Starch has an unbranched component. Lecture notes for AS Level Biology by Mohammad Rashid. Chat the cell number 03004610455761. 210. Which statement describes how the molecular structure of starch is suited in its function? The final amylose/amylopectin complex is insoluble and does not affect the osmotic properties of the cell. 211. Which feature of hemoglobin makes it a globular protein? It has hydrophobic groups on the inside and hydrophilic groups on the outside. 212. Which features of collagen result in it having high tensile strength? 1. Covalent bonds form between adjacent molecules. 2. Each three-stranded molecule is held together by hydrogen bonds. 3. Every third amino acid in the polypeptide is small. 213. HIV protease is an enzyme produced by the HIV virus. 2 identical chains of 99 amino acids form the enzyme. In each chain amino acid 25, 26 and 27 in the sequence form part of the active site. Which orders of protein structure controlled the shape of the active site? Primary, secondary, tertiary, quaternary. 214. Which statement is true for cellulose but not true for protein? It is synthesized from identical subunits. 215. Which combination of bond types correctly shows the weak and strong bonds that hold a molecule of protein in shape? Types of bond: hydrogen, weak, disulfide bond, strong, ionic bond, weak. 216. Lactose is a disaccharide present in the milk of mammals. How will a baby mammal benefit from having this sugar, instead of monosaccharide in the milk? Hydrolysis of glycosidic bond gives a gradual release of monosaccharide. 217. Which sequence correctly identifies the change in colors during the Benedict's test? Blue, green, yellow, brown, red. 218. When a peptide bond is formed, which statement is correct? 1 amino acid loses a hydroxyl group from its carboxyl group. 219. What is general formula of starch? C6H10O5, N220. A number of different types of bonds maintain the structure of proteins. These include disulfide, hydrogen and ionic bonds, as well as hydrophobic interactions. Some of these are stronger than others. Which are the strongest? Disulfide bonds. 221. How is the shape of a polypeptide chain maintained when it is coiled into an alpha helix? Hydrogen bonds. 222. Which of the following is a polysaccharide present in human muscle? Glycogen. 223. When solutions of dilute sodium hydroxide and copper, 2-sulfate, Biuret test, were added to an unknown substance, a purple color was observed. This test indicates the presence of which food in the unknown substance? Peptide. 224. What is the function of iron in the hemoglobin molecule? It binds oxygen to the hemoglobin molecule. 225. The movement of water through the vascular tissue relies on which property of water? Strong cohesive forces between molecules. 226. At which levels of protein structure do hydrophobic interactions occur? Tertiary and quaternary. 227. Which type of reaction takes place when starch molecules are converted into reducing sugars? Hydrolysis. 228. What will break an ionic bond between amino acids? pH change. 229. Bonds are formed by condensation in cellulose, sucrose, and hemoglobin. What are the names of these bonds? Cellulose, glycosidic, sucrose, glycosidic, hemoglobin, peptide. Lecture notes for AS Level Biology by Mohammed Rashid. Chat the cell number 03004610455762. 230. Which type of bonds hold the tertiary structure of a protein molecule? Disulfide bond, hydrogen bond and ionic bond. 231. Which property of water minimizes temperature changes in cells and organisms? Specific heat capacity. 232. Which levels of protein structure are demonstrated by hemoglobin molecule? Primary, secondary, tertiary and quaternary. 233. Which term describes the type of bonding responsible for stabilizing the secondary structure of a protein? Hydrogen. 234. Certain insects are able to stand on the surface of ponds. Which property of water allows them to do this? Cohesion between water molecules. 235. What is the name of bond joining glycerol and a fatty acid in the formation of a monoglyceride? Ester bond. 236. What are features of glycogen? Do not have nitrogen and structural role in the cells but have branched molecule. 237. During the production of apple juice, enzymes are used to break down the components of the cell walls. Which carbohydrate will be produced by this? Hydrolysis. Glucose. 238. Which substance contains carbon, hydrogen, oxygen, and nitrogen? Collagen. 239. How many heme groups are there in one molecule of human hemoglobin? 240. Which bonds the last to break when an enzyme is heated? Disulfide. 241. The diagram shows a molecule which is an important component of living organisms. A forensic sample is tested for the presence of a polymer of this molecule. Which polymer is tested for? Protein. 242. What is the general formula of a monosaccharide? CH2ON.